Welcome to this video lecture, human insulin production. Okay, so in this video lecture we will see what is human insulin, what are the function of the human insulin, and why we need to produce this human insulin. So this human insulin, if we see, it's actually a hormone. Okay, so human insulin is a hormone. So this hormone is very essential to maintain the blood glucose level. Okay, so this insulin is a hormone which plays a very important role in maintenance of blood glucose concentration. So if the blood glucose concentration is more, then what happens? Insulin act on the insulin induce the cells to uptake the blood glucose. So here this is the introduction. So insulin is a hormone made up of protein. So it is secreted by beta cells of islets of Langerhans present in the organ pancreas. So it is very much essential for the transport of glucose into the cell. And how it transport glucose into the cell means this insulin goes and binds with the insulin receptor in the cell, which in turn opens up the GLUT4 receptor, so which accepts the glucose from the outside environment and pass into the cell. Then this insufficient secretion of insulin leads to hyperglycemia, which means the higher blood sugar level. So this higher blood sugar level, it is known as a condition called a diabetes. Okay, so this diabetic condition is known as the insulin dependent diabetes, because the glucose concentration here, it's mainly risen due to the non-availability of insulin. So it is mainly because of the insulin dependent diabetes. Okay, so this is the very short introduction about insulin, and in the forthcoming slides you will see the structure and function of the insulin and its production, how this insulin produced using the recombinant method and all. Okay, and if you see this structure of the insulin, so the structure of the insulin, if we see, as I told earlier, this insulin is a proteinaceous hormone, which means it is made up of protein. So here this insulin contains two chains. So there are two chains of insulin present in here. So one is the insulin A chain, and the other is the insulin B chain. So insulin A chain and the insulin B chain. So we will see one by one. And this insulin A chain and B chain, if we see, it's held together. It's held together by the disulfide bridges. Okay, so this insulin A chain and the B chain is held together by the disulfide bridges, and for its activity, so for insulin to be very much active, it needs both the chains. So insulin A chain and the insulin B chain. So if insulin A chain is available alone, means then what happens? It will not be a functional insulin. Similarly, if insulin B chain is available alone, then it is not a functional insulin. So the molecular weight of the insulin is about the 35,000 daltons. Okay, and as I told just before, it's made of two polypeptide chains. So the A chain and the B chain. So A chain contains 21 amino acids and B chain contains 30 amino acids. So these two chains are linked by the disulfide bridges. And coming to the function of the insulin, so if you see the function of the insulin, it reduces the blood sugar level. So it reduces the blood sugar level, and it promotes the glycogen metabolism in livers and muscles. Okay, so it promotes the glycogen metabolism in liver and muscles, which means what is glycogen metabolism? Means whenever the body needs glucose, then at that moment what happens is the glucose is stored in the form of glycogen. So the stored glycogen energy, this stored glycogen gets break down and it releases the glucose molecules. That function also will be regulated by the insulin hormone. And next, if you see, it also prevents the breakdown of the cellular proteins. So this is the function of another function of the insulin is it reduces the breakdown of cellular proteins, and Moreover, it maintains the blood glucose level. Okay, so this is the diagrammatic representation of human insulin. So here chain A contains 21 amino acid which is situated below, and chain B contains 30 amino acid which is situated above, and both the chains are held together by the disulfide bridges. Okay, and if you see the chain A, it's the amino acids sequence start from glycine to asparagine, and if you see the chain B, the amino acid sequence start from phenylalanine and ends in threonine. So the amino group in phenylalanine and the carboxyl group is contains threonine in chain B. And okay, so now coming to the genetic manipulation of E. coli for insulin production. So as we know, this insulin hormone is a very essential hormone uh, for treatment of diabetes, right? So in order to produce this insulin in a larger quantity, so earlier what they will do means they extract the insulin from the pancreas of pig and other large animals. But now with the advent of the RDNA technology, it's very easy to produce insulin via the bacteria called E. coli. Okay, so the first thing we have to take two plasmids. So in two individual plasmids we have to take in a plasmid, one plasmid we have to take insulin gene A. The second plasmid you have to take the insulin gene B subunit. Okay. So if you see the plasmid, it contains antibiotic resistant gene, promoter gene, beta galactosidase gene, and insulin gene A subunit in one plasmid, and in insulin gene B subunit in another plasmid. So this insulin gene, it is located downstream to the beta galactosidase gene. So whenever this beta galactosidase gene gets expressed, this insulin gene also gets expressed. So this is the scenario of how we can produce the insulin through E. coli. So once a plasmid is ready, then we have to transfer the plasmid into the E. coli host, okay, by means of transformation. And when the E. coli is cultured in the media, this E. coli cells, there is a plasmid insulin gene present in the plasmid gets expressed, and it forms the beta galactosidase insulin A fusion protein in one cell, and in another strain beta galactosidase insulin B fusion protein in another E. coli host. Okay, so now we can purify the fusion proteins. So since it is available as fusion proteins, but we need only insulin A and insulin B chain. Okay, so that's only we need. So we have to cleave the insulin A chain and the insulin B chain from beta galactosidase using some chemicals. So one such chemical is called cyanogen bromide. So using cyanogen bromide we can cleave the A chain and B chain respectively. So once the A chain and B chain is available, we can once the A chain and B chain is available, we can mix it together to make the active insulin. So this active insulin is now it becomes a functional insulin. So this insulin which contains insulin A and insulin B polypeptides now becomes active, and it's ready to use as for treatment. So the picture shown here is called humulin N. This is a prepared formulation of this recombinant insulin produced using the E. coli. Okay, so this is how the insulin is produced from E. coli by means of the genetic manipulation. So from this we can know that this RDNA technology or recombinant DNA technology provides a valuable tool for production of this human proteins of therapeutic interest in a larger quantity. So definitely when compared to the conventional methods, this genetic manipulation method is really a very I mean very um, essential and very useful application in order to produce this insulin in a larger quantity. Okay. So after this we will see the procedure. So this is the diagrammatic representation of the procedure, and after that we will see some step by step procedure how they construct this plasmid and how the transformation is done, and how the fusion protein is produced, and how they purify the fusion proteins. Those things we will see. Okay, this is the procedure. This is the procedure uh, how the insulin is produced. So DNA fragment coding for insulin A and B chain was cloned and expressed separately in different E. coli cells. So one E. coli produces insulin A chain and another E. coli produces insulin B chain. So both the chains are purified separately and treated with cyanogen bromide, okay, to clear the beta galactose from insulin proteins. So this insulin A and B chain mixed together to form disulfide linkage. So now we get the biologically active insulin molecule. So this insulin produced through this kind of genetically manipulated E. coli can able to produce nearly 1 into 10 power 5 molecules of insulin in a week. Okay, which is a very larger quantity when compared to the uh, conventional methods. Thank you.